Good evening, everybody. Welcome all of you to CSIR National Geophysical Research Institute for the 88th CSIR Foundation Day celebrations. Good evening, madam. We welcome you here with us. In the morning, we had a virtual interactive session with the registered participants from all over the India. And now, as a part of these celebrations, we are having a memorable talk by an eminent personality. To follow the procedure, first, we would like to have the CSIR song played among us.
We are not able to hear it.
वैज्ञानिक औद्योगिक अनुसंधान करते हैं इस देश का कल्याण। आओ हम सब हाथ बढ़ाएं, इस भारत का मान बढ़ाएं, इस भारत का मान बढ़ाएं। भविष्य विज्ञान भारत का देखे सपने दृष्टाओं ने साकार किया, उन सपनों को साकार किया, उन सपनों को परिषद के सब स्रष्टाओं ने, परिषद के सब स्रष्टाओं ने। समय समय पर साधना करके, अणुवीक्षण से आरंभ करके, मन के संघर्षों को जगा के, मौलिक धाराओं को उगा के, मौलिक धाराओं को उगा के। लेकर नवीन आविष्कारों को सर्वजनों के सम्मुख जाए, विश्व विजय का केतन लेकर, विश्व विजय का केतन लेकर विज्ञान का विस्तार करेंगे, विज्ञान का विस्तार करेंगे, विज्ञान का विस्तार करेंगे।
Now I request Dr. V.M. Tiwari, Director, NGRI, to give the opening remarks of today's program and introduce the chief guest of the celebrations today. Over to you, sir.
Thank you, Tanvi. The 80th CSIR Foundation Day Speaker, Professor Chandrima Saha, former directors of CSIR NGRI, Professor Harsh Gupta, Professor D.P. Dimri, other distinguished guests, I see Professor R.N. Singh, Professor Singhi, and also Professor D.M. Banerjee, many other colleagues joined through online, CSIR NGRI family, and students. Let me begin by wishing the entire CSIR family many happy returns of the day. It's the 80th birthday of CSIR. CSIR has always been in the forefront of science and benefiting society through scientific and technological interventions. Often said, like mother, like daughter, So the CSIR NGRI, which is celebrating its Diamond Jubilee, has been immensely contributing to the geophysical sciences and their application for societal and economic benefits. In the recent past, CSIR NGRI has put in efforts to solve community problems while focusing on the basic research. Some of the fundamental science issues like accretion of the continent during early history of the Earth, lithospheric structure and earthquake genesis in the Himalayan and Andaman regions, seismic hazard scenario generation due to the Himalayan earthquakes, and many other science issues which are being globally debated are being pursued at NGRI. In recent time, we have also embarked on two major projects of the strategic importance. One is for the exploration of uranium through supports from Atomic Energy Directorate, Department of Atomic Energy, and another mapping of aquifer in the arid region of Northwest India through Jal Shakti Ministry. They are the ever largest projects CSIR NGRI had in the past. Incidentally, today is Daughters' Day, and combining it with the 80th CSIR Foundation Day, we are honored to have a CSIR alumnus, a worthy daughter of India, Professor Chandrima Saha, who made mark in the scientific landscape of India and global biological community. We are also celebrating Azadi Ka Amrit Mahotsav, and it is very pertinent and befitting to mention that Professor Chandrima Saha mother went to jail for pulling down the British flag. So there cannot be a better person today to talk with us who is one amongst us few decades back at one of the CSIR laboratories. Apart from her illustrious family background, Professor Saha has several firsts to her credit. She was a vice captain of West Bengal's first women cricket team. So those who see IPL must be wondering that there is a woman player who also became the first cricket commentator for All India Radio. And today she is the president of the Indian National Science Academy, New Delhi. We all know that CSIR Jigyasa program is making a big presence in the common man and also reaching to the school children very much in the different parts of the country. Similar to that, Professor Saha started a concept called Vigyan Setu, and that also became very famous. Why I narrate all this? Because we have today not have only a very eminent researcher, but also a person who has made her marks in the different parts of our life. Her research is mainly focused around what causes the cell death. And this is very important in the way that cell death is very fundamental to our bodies. She had been in forefront in conducting the extensive research about Leishmania parasite which causes Kala-Jar. So one side, while she pursued a fundamental research of understanding about the cells, she had addressed the bigger issues of humankind like the diseases of parasites. The Professor Saha is graduated from University of Calcutta and obtained her PhD while working at Indian Institute of Chemical Biology, one of the CSIR laboratories, and also worked at several places in United States at postdoctoral research like University of Kansas and Population Council in New York City and National Institute of Immunology, New Delhi, where she worked as a scientist and rose to the level of director. She has served in several committees and councils. It's very difficult to name everything. It's a very long list, but to tell that in all three academies, she had served in one way or other. The another thing which she has been very much contributing through her knowledge is serving in the councils of IITs, ICMR, and many biological laboratories. He is a recipient of several awards and honors, and remains recent amongst them had been the Shanti Swarup Bhatnagar Medal from INSA in 2019. She is fellow of all three distinguished science academies of India and also the fellow of the World Academy of Sciences, Trieste, Italy. She had JC-based fellowship. She has many other awards like Pushpa Sri Machari Foundation Day Oration Award. She got the DP Burma Memorial Lecture Award, and so on. I will not come between Professor Saha and all of you and request Professor Saha to deliver the 80th CSIR Foundation Day lecture on her chosen topic, looking into the life, the beauty of cellular struggle. And I would close with thanking her for her kind gesture of agreeing in a very short notice to deliver CSIR Foundation Day lecture. It's over to you, Professor Saha.
Thank you very much, Dr. Tiwari, for a very generous introduction. And you know, I mean, I don't know how you got all these details, but you know, thank you very much. And I would like to greet also Dr. Harsh Gupta, Dr. D.M. Banerjee, Dr. Dimri. I can't see all, and other distinguished guests are there. And it is indeed a great pleasure to speak at this 80th celebrations. And I'm not only an alumni of CSIR, I'm currently also with the Indian Institute of Chemical Biology as J.C. Bose Distinguished Chair Professor. So currently I'm serving CSIR. So this lecture, actually, it is meant for a broader audience because this is a very different institute. And so if experts are there, my apologies to them, but I have kept it in a very general terms. And can I start sharing my slides now?
Yes, madam.
Yes.
You have to go top right, and there is an arrow.
Yeah, put the. They have to allow me to share.
Can we?
Sir, it is already there, it seems.
Okay,
Sir,
I have. It says screen share.
Yes, ma'am. Ma'am, you have to open the presentation and then it will come on sharing mode.
Okay, I have the presentation open. Can you see my presentation?
Not yet, ma'am.
Okay, let me see what happened. enter screen share. No. Can you see now?
Yes, ma'am. Ma'am, you can you put it in slideshow mode? Yes.
Yeah. Now, is it
Yes, ma'am.
okay?
Yes, ma'am. It's okay.
Okay. So when we talk about biology of cells, the very fundamental units which we are a conglomeration of, we have to remember the governing principles that runs lives. And for that, we are very grateful to two gentlemen. One is Charles Darwin, which about whom all of you have heard, and also Gregor Mendel. These were the two most influential 19th century founders of modern biology. When Darwin observed the finch beaks and developed his theory of selection, and Gregor Mendel, while studying peas, he also developed the theory of the heredity. But what happened with the Gregor Mendel is his theories were kept, people did not believe him, and they were, for a long time, they were not recognized, whereas Darwin was recognized. And interestingly, both Darwin and Mendel were contemporaries in a way. While Darwin did not know about Mendel's work, Mendel knew about Darwin's work. And this we have come to know because with Mendel's papers, there were Darwin's papers also. So Mendel was actually reading Darwin's papers. So both were very different characters. And in 1831, Darwin embarked on a 5-year journey around the world on a ship called HMS Beagle. And Gregor Mendel started in 1856 working in the garden of a monastery in Moravia. And so Darwin developed the theory of evolution by natural selection, where he surmised that individuals show variation of physical characters, and the individuals who are most suited to survive in that particular environment will survive and given enough time, and the species will gradually evolve. And Mendel developed principles of inheritance that describe the transmission of genetic traits before anyone knew that genes existed. So now Darwin's theory of natural selection actually lacked an adequate account of inheritance, making it logically incomplete. Now, once the particulate basis of genetics was understood, it was seen to allow variation to be passed intact to new generations. So this was the theory that he observed while he was taking his trip in HMS Beagle over some of the archipelagos. And he said that evolution could then be understood by this process of change in frequencies. Subsequently, with Mendel's theory coming in, evolutionary genetics developed as a central part of biology. So both Darwin and Mendel are responsible for our understanding of much of our biology, and also without Darwin's theory of evolution, nothing really makes sense. So the humans evolved from about 200,000 years ago, and the modern man walked out of Africa in about 70,000 years ago. So while humans evolved, from the ape-like ancestors to the modern man, they also acquired many pathogens. Many pathogens started infecting humans. And if you see, this is an example of a bacterial growth at 15-minute intervals. You can see the bacteria are actually dividing very fast. So a human's life and a bacterial life, if you compare them, bacteria virus, they can divide much more faster than us and have many variations and many mutations that can happen. And those favorable mutations can be maintained for the species to survive better. So with those, what happened with those is that there is a race against pathogens that started happening with human evolution. With the selective forces, Darwin's selective forces, actually what he predicted, acted on pathogen pathways so that it could survive better in the host. And selective forces also worked on host pathways so that it can eliminate the pathogen effectively. So this was this war that was going on during evolution, and eventually it depends. That war is still going on, and with the coronavirus pandemic, you know how the war is going on. And I'll show some examples where how it shows that Darwin's theory still is valid and has to be studied in order to understand how the pathogens are actually infecting us and creating successful infection. So when humans were solitary creatures in the sense they were hunter-gatherers, they were just killing animals, eating them, and sort of solitary, or maybe in groups of 2 or 3, the infections were less. But when they started building groups and made villages, the infections started spreading because the humans came in close contact and started spreading all over. And that is why when the world population grew from 200,000 to today's 7 billion, actually the infectious agents also grew in number, grew in variety, and started affecting, and the pandemic started happening. So if we look at the pandemics in history, what we see is that the earliest evidence is smallpox, which dates back to about 3rd century BCE, caused by the variola virus. And this very smallpox was effectively eliminated from the world by systematic vaccinations. One of the reasons that experts think why smallpox was eliminated was because it did not have an intermediate animal reserve. So animal reservoirs are very difficult to eliminate. Then came the Spanish flu. The Spanish flu was during 1918 and 1919. It came to India also. We had a lot of deaths, and it was caused by the H1N1 influenza virus. So, and the most fatal of the pandemics was the Black Death or the plague. So the plague is caused by a bacteria called Yersinia pestis, which is carried by rat fleas. And so the death toll in the 14th century was about 200 million. And it came back again in the 19th century and caused a death toll of 12 million in 2 places, mostly in China and India, and the other world counting is separate. So, and other pandemics are HIV, for which we still don't have a vaccine. The SARS vaccine is in development. The MERS, there is no vaccine. And this is the Middle East respiratory virus. And so in 2014 to 16, we had the Ebola. Ebola is a very, very deadly pathogen, and there is only one vaccine. Now, Ebola vaccine, not so much force was given because the Ebola virus actually was confined to a very small area in Africa. So it did not get that much attention, but it's a very, very deadly virus. Then, of course, we have the coronavirus pandemic now, where this figure has actually changed and increased. So these pandemics came all across history, and vaccination seems to be a very, very effective means of eliminating the pathogens. Sometimes we're successful, sometimes we're not. If you look at this schematic, you see that there are emerging and re-emerging infectious diseases that are present now. So we have what we call newly emerging pathogens. We have re-emerging or resurging pathogens, and we have deliberately emerging pathogens in which anthrax has been named, and not any others till now. So there you can read the names, that there are so many of these new viruses, new bacterial diseases, fungal diseases are coming in. So for the re-emerging diseases, they can be previously unknown infectious agents or pathogens that evolved over time. And known agents that have been spread to new geographic locations, suppose it goes from India to another location, which has actually happened because travel has increased, and because of so much travel all across the world, diseases have been spreading very effectively. Known agents whose role in specific diseases has previously gone unrecognized. We didn't realize these agents were there, but later with these new molecular biology techniques, it's now known that these agents are there. And also re-emergence of pathogens whose incidence had significantly declined in the past, but it has reappeared again and is coming back. So once we eliminate something, it can again come back and strike us because we don't know where they are actually lodged. So this is a very recent happening between 2011 and 2017, where you find that the cholera, the Zika virus, the meningitis, West Nile fever, all of them are coming in very small amounts. They're happening across in very small amounts, but they can at any time increase and create a pandemic. So these have been less because of effective treatments and some measures to control these, to control the spread. So this is very recent. Now, in the past, people did not know what were causing the diseases, and it was called a miasma, meaning bad air which is falling upon the humans and it is causing the diseases. So many questions were asked, like why is there sickness? Why only a few people are sick at a time? Why not everyone? And why so many people are afflicted sometimes? Why some recover better and why some succumb? So these were many relevant questions that were asked. Now, the germ theory actually originated very early in the Middle Ages with very famous physicians like Ibn Sina, who actually predicted that there was something outside the body which is coming and causing the disease, although they did not know the type of things that are causing that. And Fracastoro, an Italian scientist, or he was a scientist philosopher, who actually predicted that there are some seeds which are coming and causing the diseases. But there are many things that happen, but it's not possible to cover all those. But actual breakthrough came when Antony van Leeuwenhoek, who was a draper, a Dutch draper, who actually created the microscope. So he had a very small contraption of a microscope, where if you put the sample here, you can see the living world under this lens. So Van Leeuwenhoek actually described small animalcules, meaning motile germs or motile agents that may cause disease. But he could not link disease to these, but he did see these motile characters. So Robert Hooke, who became the president of the Royal Society, actually made some higher grade microscopes which were more powerful and looked at the slices of corks, and he could see that there were little holes in the corks. He called them cells. The reason is it reminded him of cells occupied by the monks in monasteries. So he called them cells, and since then the cell has become to be known as the cell. Actually, it was due to Robert Hooke. Now, cells we know with very high power microscopes, we can see the cell, its cytoskeletons, it's the organelles inside. So cells are now very easily being looked at. So some very interesting facts about cells in our body is that total cells in our body is about 50 trillion. And every day in our body, about 10 to the power 11 cells are formed. But at the same time, about 10 to the power 11 cells also die per day because you have to make space for the older cells. So we actually, as we speak, our cells are dying and are being born. So within a year, the amount of cell a person loses through cell death is almost comparable to the entire body weight. So by the end of the year, we are a new us in the sense that our cells are being, cells are being refurbished. But it doesn't happen in all parts of the body. Selected parts have more cell death, selected parts have more cell growth, like that. So it is an interesting thing that we have so much death in us. Even during the child being in the mother's womb, cells would die. And so a person, when he's born, he's already died, his many cells have died. So cell death is a very important aspect, and I have worked on cell death most of my life. But today I'm actually not talking about cell death because it's a very complicated subject. So now I just wanted to show that this is a very healthy cell. These are the gift of the very, very powerful microscopes that we have now. So this is a healthy cell which is dividing, and this is a cell which is dying. And so it is dying, and it dies through, this is a very controlled form of cell death, where it dies by producing little blebs. These little blebs do not break, and so the cellular contents cannot spill out. And the cell dies in a very organized manner. But this cell death can be due to many reasons. One is there could be hypoxia, there could be ischemia, by which the cells are not getting their supply of oxygen and food, and they can die because death can be initiated. Then mechanical trauma, like any mechanical trauma of say a shear, a cut or something, cells would start dying there. Then chemical trauma like pesticides, poisons, drugs, they can cause cell death. Infectious agents are one of the foremost agents that cause really cell death in many cases. Then there is immune reaction. So immune reaction can save us from many diseases, but immune reaction, if gone awry, can really cause huge problems in the human body that we are seeing with the coronavirus patients, where overt immune reactions are actually causing death. Then genetic disorders can cause cell death, inherent like the lupus disease. And then nutritional imbalances also can cause cell death. So there are a variety of reasons that can cause cell death. And other than the normal death we have, the normal cells that are being replenished, they're dying, the old cells are dying, they're being replenished by younger cells. So other than that, these are the kind of assaults that can cause cell death. This is a photograph of a cell which is infected by coronavirus. You know that the coronavirus attaches by the spike protein, and it's all over the cell. If you cut a section, you will see them inside also. This is shown by scanning micrograph, and this is the transmission micrograph, where you can see they are within specialized bodies, either phagosomes or endosomes. They are within the cells. Now, see, a very virulent pathogen has this hazard, because as it infects the body, the first thing the body does, it tries to kill. So, so it is also under this hazard. It can also kill the host if it proliferates too much. And therefore, cell death, in a way, cell death represents a very primitive and highly evolved innate immune mechanism that pathogens come in, the cell tries to kill it. And it maybe continuously need to be able to manipulate it. Now, if you look at this diagram, you see that when the pathogen comes in, the cell successfully kills it through innate immune mechanisms or the inherent mechanisms that are in the cell itself, then the cell is saved. Now, if the cell is unable to kill the pathogens, what it does is it inflicts cell death on itself. This is actually cell suicide, and it dies by taking the pathogens with it, which is being cleared by the macrophages, and the body is cleared of the pathogen. Now, these pathogens are very smart. They actually can subvert host cell death. They are viruses, mostly bacteria and viruses are very good at subverting host cell death. So this is actually a diagram which shows the cell death pathways. One is the cell death receptor bearing pathway with death ligands are there, and another is through the mitochondria that the cell death is executed. So all of them are executed by effector caspases. But I put this diagram because I wanted to say here is that whether it's the ligand for the death receptor, whether it's the activation of caspase 8, whether it is the the mitochondrial proteins, everything can be mimicked by the viruses and also by bacterial proteins. And that is how they actually subvert host cell pathways and survive in the cell. Because the virus has to have a cell to survive, the bacteria mostly has, they can stay extracellular niche also, but they can survive within the cells. So if they have to survive within the cells, they have to subvert the host cell suicide pathway. They have to subvert all the metabolic activities of the cell so that the cell doesn't die and it also doesn't die. So it's a very complicated procedure, and we have learned many, many things about this. So now, in case when we come to immunity, it's the response to infection is very very important. This was shown very early by Elie Mechnikov, who's a Russian scientist, and he got the Nobel Prize in 1908, and he showed by taking a transparent jellyfish and putting a rose thorn in it, the cells were migrating towards the rose thorn as if to throw it out, as if to push it out of the cell. So this was the first description of the immune reaction. So the immune reaction is carried out by certain peculiar cell types within the body, and by now you must have heard these names. One is the innate immune reaction, where dendritic cells, macrophages, and neutrophils take part. They first try to kill the pathogen by reactive oxygen species or other mechanisms that are with them. And if they cannot, then the help of the adaptive immune system is brought in, where helper T lymphocytes interacts with macrophages and dendritic cells. And the macrophages and dendritic cells, what they do is they chew up the pathogens and take their proteins, put them on the surface. And that is with what the helper T lymphocytes reacts and then sends the message to B lymphocytes, which differentiates into plasma cells, that you have to generate antibodies, and they do generate antibodies. And also helper T lymphocytes talks to the cytotoxic T lymphocytes, which differentiates. They can come and kill the infected cells. So this is in very, very brief what the immune system does when a pathogen strikes the body. Now, we can now see life up close because of this high power microscopy. Here, this is a macrophage, and it is engulfing Streptococcus bacteria. So what it does is once it engulfs it, it takes it within some structures called phagosomes. And once it takes into the phagosomes, the cellular lytic enzyme bag called the lysosome actually comes and fuses with it. As the lytic enzymes of the lysosome chew up the pathogens, they are either discarded out, the raw materials are used up, or they are put on the cell surface, as I said, for the T cells to react. So this antigen presentation is very, very important. The antigen presentation, again, is a very complicated thing involving the HLA the proteins on our cell surface. So this is another very important aspect of immunology that one has to study, that how the antigens are being presented. So in case of the coronavirus also, we haven't got that much of time to actually do that kind of studies, that what are the HLA profiles of people who get more infected, but that will surely give us some good leads. Many years ago, when the sensitive reactive oxygen species sensitive dyes came, we actually showed that in the phagolysosomes, where the lysosomal enzymes were reacting on the pathogens, also the reactive oxygen species were being produced. So this is a lysosome and this is a phagosome. This is the lysosome with the reactive oxygen species in green, and if you overlap them, you see the same places where it is being produced, the reactive oxygen species. What happens is the NADPH oxidase lodged on the surface of these phagosomes, they actually convert oxygen to superoxide, and superoxide dismutates into hydrogen peroxide, which then generates highly reactive hydroxyl ions. They combine with ROO, peroxyl radicals, and in the presence of nitric oxide can form again peroxynitrate, which is highly reactive, and they can dismantle any pathogens if required. So this then we also went ahead and we also showed at that time that within the phagosomes, the pathogens are being killed and they die through the process of apoptosis, which is a neat process of disposal of cells. And these are the macrophages which have no reactive oxygen species, and the pathogens are not dying. But here, the oxidative stress has been given and they start dying, and the green marker is actually showing apoptosis. But the nucleus of the host stays red. So these are confocal micrograph, and they're showed in optical sections. So now if you look at antigen presentation by dendritic cells, I said macrophages, I said dendritic cells. So dendritic cells, this is a dendritic cell. This is a scanning micrograph of a dendritic cell. These are all pseudo-colored, actually. So this is a T cell. So the T cell is interacting with the dendritic cell and reading the antigens which are put on the surface. So if you look at the region of the reading, you'll see it's a very, very complex kind of reaction where the MHC molecules on the surface of the dendritic cell is presenting this antigen to the T cell receptor so that the T cell can get activated and go to the B cell and cytotoxic T cell to tell them to actually mount an immune response. So this we have now been able to see through the microscope and through molecular biology studies. So again, this is a macrophage which has a T cell sitting on it, and it is eating up the Streptococcus bacteria. Once it does that, it recognizing the antigen, it actually reacts with the B cell, and the B cell starts producing antibodies. So this is the principle. This is actually the principle of vaccination. When we are given an antigen, it is picked up by the macrophages. Antigen is presented, and the T cell now gives the message to the B cell, okay, antibodies can be provided. And one important thing that happens at that point is that the B cell memory, memory B cells are produced. So the plasma B cells secrete the antibody when memory B cells that are produced, that are kept in the lymph nodes. And if in future you get infected, then those memory B cells will come and start producing the antibody. So this is the principle of vaccination. And we have known many things about vaccinations now. And as I said, it is one of the most important mode of eliminating disease. So I'll just give you some examples how Darwinian theory of evolution works. So here also in this virus, you see the spike proteins. You are very familiar with this photograph by now. The spike proteins in a very structural biological terms would actually, this is the virus body, this is the spike protein, one single spike protein. Actually, it's a trimer, and it actually binds to the ACE2 receptor on the surface of the human cell. So this is the binding point. So most of the mutations would occur in this protein, in the spike protein, because the spike protein is the one which interacts with the human cell with the ACE2 receptor, and maximum variation can be achieved when mutations occur in this particular spike protein. Now, it is interesting that just the binding of spike protein to ACE2 receptor is not enough to cause the infection. So there is an enemy within, which is called TMP-RSS2, which actually creates a cleavage on the spike protein so that it can bind with after the ACE2 receptor, it can bind with the membrane and the virus can get into the cell. The similar thing is also done by another protein called furin. The furin cleavage site here in the spike protein also causes this binding to the membrane and helps the virus from getting in. So this is how the virus actually gets in. Now, there's a statement by Parvez Hudba that every hope of dealing with today's rogue virus rests squarely upon Darwin's 200-year-old discovery of the principle of natural selection. So if there is a mutation in the spike protein, if it is good, then for the virus, it will survive. Most of the mutations are not very radical, or if it is very lethal, then it is actually discarded because the virus actually doesn't want to kill its host, but it does so through collateral damage. Now, with organisms with genes better suited to survive in a given environment are selected. So who is the fittest? If the host can kill the virus, the host is fitter than the virus. And the virus is getting fitter by getting all these mutations during the time it circulates within the population. So in case of immunity, what happens is it's actually like a double-edged sword. Because in one hand, it's helping the host to fight infections. And on the other hand, it can cause significant damage in the form of autoimmune diseases. The reason is that when in the innate immune system is activated, cytokines, which are proteins known to cause inflammation, are secreted. So they cause an inflammation because at the inflammatory reaction, all the all these defensive cells can come in and kill the viruses or the bacteria. But if you can't bring down that inflammation, which is necessary after healing, that the inflammation has to be brought down. Sometimes it isn't done, and thereby there are casualties in case of the coronavirus, where it is very, very severe. So I wanted to give one example of a mutation. So there is the coronavirus in the first, when it appeared, it came with a D residue at 614 of the viral spike protein. Now by April of 2020, a single mutation in the genome was found that converted D to glycine. Now when we see the sequence of the viruses, the D variant has been all but replaced by the G variant. Now globally, you will find only the G variant. Why? Because this G substitution provides some sort of selective advantage to the virus. It transmits more readily than the D variant. The number of functional spikes on each viral particle is 5 times higher if there is a substitution with glycine. And patients infected with the G variant also has higher viral loads in their upper airways than that was found with patients with D. So this actually shows that why it is very important for the virus itself to get a substitution which will be advantageous to us. Higher viral loads, if found in the airways of patients, it could lead to increased transmission. So that is also very important. So like this, there are many mutations that has happened. Some of them are good, some of them are bad. But like the N438K substitution, it emerged and circulated widely in many European countries. And where it was, asparagine at the 439th site was replaced by lysine. Now this enhanced the binding affinity for the ACE2 receptor. So it was of advantage to the virus. So this binding energy of N439K was very high, and that of the comparatively with the wild type complex. And the mutated virus formed more hydrogen bonds than the wild type complex. So you can go to that level to see what is actually causing this increased binding. It was the hydrogen bonds. And the replacement of asparagine with lysine actually formed a new salt bridge in the complex with ACE2 receptor. So which could increase electrostatic interactions. So these details were all realized by basic research, and that is why we are slowly able to counteract the virus in many ways. But what is important is, and what is worrisome is, like the Delta variant that we have now, it is reducing the neutralizing activity of the polyclonal antibodies present in the sera of the people who have actually recovered from infection. Interestingly, there is a study in Israel where they have checked for antibody titers in people who have been vaccinated. And in about 6 months, this was mostly aged population, but in about 6 months, the antibody titers have gone down. So, but they did not check the T cell mediated immunity, or that should have been checked, but maybe it's difficult to with the cohorts. So, but they did check the antibody titers, and since the antibody titers are going down, they have now recommended a third booster. But there is this controversy now that when all the people in the world has not been able to get the vaccination, how come you can recommend the third booster? But that's a policy of the country. So the mutations, as I said, are highly deleterious, and which are highly deleterious are rapidly purged. High effect mutations that contribute to virus adaptation tend to be less, and a small minority of mutations impact virus phenotype in a way that confers a fitness advantage. Again, Darwin's theory is coming back, and that fitness advantage actually helps them to infect more. And it affects the pathogenicity, infectivity, transmissibility, and antigenicity. And so then there is an increased infection, and the disease circulating through the population actually increases. There was, as per estimates, there were 2 mutations per month in the global population. it has, so far as I know, it has not increased, but it is about 2 or 1 per month in the global population. So this virus is getting a lot of mutations. And what we are, this is the vaccination profile all over the world, and India stands here with about 45% of population being vaccinated. One thing is for sure, that it will be almost impossible to get herd immunity. Herd immunity is something by which most of the people, vaccinated people, will not spread the disease. So so unvaccinated people within the population will not get the infection. But in order to get herd immunity, you have to have most of the people vaccinated, say about 80%. So we have not reached near 80%, but even if we get 80%, what will happen to the people whose immunity are waning and they require to get a third booster? So this is a very complex question, although India has done a very good job by vaccinating, by a very organized vaccination program. But still, it is difficult to get a herd immunity. And so in the absence of that, we have to be very, very careful. So for the future, science has discovered much about pathogen biology and made extraordinary strides on the development of vaccines and drugs. If you look at history, which I showed you about the pandemic, that we have understood pathogen biology a lot. We don't know, it is true that we don't know much of it. And we have got drugs, we have got vaccines, which have eliminated some diseases, which have not eliminated some, like one other than smallpox is polio. For polio has also been almost eliminated. So the funding, because all of these stand on fundamental biology, the fundamental biological information was available at the beginning of the coronavirus pandemic. And that is why vaccines were produced so quickly and drugs were not that successful. But anyway, whatever information we had about this virus, how it spreads, that's why the masks were imposed on. So it is very important to have basic research, and increased funding should be given by the governments all across the globe. The future will also depend on the ongoing evolution of the existing and emerging pathogens, and also the behavior of people. It is very important for the people to behave. So that is why it is essential to communicate to the people the actual facts and make them understand that they will have to behave in a certain way in order to avoid this kind of pandemic happening again. And that also depends on the decision of the governments, because we know that there are reasons why governments sometimes do not, say use masks or do not use masks. And so there has to be a communication between the government and perhaps the academies, who will actually tell the government that this is how you should go about. And we have actually brought out a white paper on this COVID-19 vaccine to sensitize the government. And for this pandemic, unless as I said about the high vaccine coverage is achieved, this stoppage of masks is premature. Hand washing, social distancing also has to be continued. And if you look at the television, if you look at the cricket matches, if you look at the football matches, people are not wearing masks, and they are all over the place. So there are incidences are happening that when people are getting the coronavirus, and now it is in a sort of, people think that we've got over it. It's not like that. It can come back anytime and bounce back. So I think we need to be very, very careful with this. And we have learned so much about the viruses, the pathogens that we encounter, but we have much left to learn. Thank you very much.
Thank you, madam.
Thank you, ma'am, for such a colorful presentation, and again about seeing virus in such a colorful mode. Now I request Dr. Kirti Srivastava, Senior Most Scientist of the Institute, to propose formal vote of thanks.
Ma'am, it was really wonderful. Professor, ma'am, can we ask you some question?
Yes, sure, you
It
can.
may be a stupid question, but is there a way to measure the T cells in my body?
Yes, yes. There are ways to measure T cells, but normally the diagnostic labs do not measure T cells because they have to be measured with a particular markers, which are very expensive, and these can be done in immunology laboratories. Actually, that is what the Israel people should have actually, when they were doing the serodiagnostics, they should have actually measured the T cells because T cell immunity is also important. And you can see some people are having very low antibody levels, but their T cell immunity may be very good.
Thank you, ma'am. Anybody else wanting to ask any question to ma'am?
Professor Kirti Srivastava, generally we don't entertain question during the foundation day lecture, but since it's a very popular, we can send all the questions to ma'am and get the replies and send to respective people.
Sure, thank you so much. Actually, I was very inquisitive after listening to ma'am. And, Professor Chandrika Saha, thank you so much for delivering the 80th CSIR Foundation Day lecture today on looking into life, the beauty of cellular biology. It was a pleasure, ma'am, to hear you.
Thank you very much.
And ma'am, you drew our attention starting from Charles Darwin to George Mendel and being responsible for the evolution of genetics. You took us through the pandemic history, starting from smallpox, we heard about it earlier, to Spanish flu, which we came to know more about it during this pandemic, to COVID and how the disease evolved and how germ theory came into existence, and how we spoke about T cells. We heard about it only during this pandemic, and then how people started saying that T cells is the only thing which would help us. So we were, like wondering how these things happen. But today, you could show us how a T cell looks, because none of us, scientists would have known how really a T cell would look. Thank you so much, ma'am, for that. And for your research on disease, which is helping the human race. And ma'am, you have so many recognitions to your credit. We are all proud of you. You are not just a daughter of India. You are a daughter of a patriotic family. Of all the women, we all stand up to you. And you have given such a wonderful talk today to all of us. Thank you so much, ma'am.
Thank you.
And I think if we have any questions, we'll send across to you.
Sure.
Thank you so much, ma'am.
Thank you. Dr. Tiwari, thank you very much for inviting me and giving me this audience. Thank you very much.
It's the last thing is left, I think, the national anthem.
Yeah.
We have to stand for.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे. Thank you, one and all.